Hello, my name is Dr. Ralf Schmid. I'm Research Director for Neurodevelopmental Diseases in the Gene Therapy Program at the School of Medicine at the University of Pennsylvania. I would like to thank the organizers for giving me the opportunity to share our progress and development of gene therapy for CDD with you today. CDD is a rare neurological disease. There are about 77,000 different rare diseases today. 80% of those diseases are genetic in origin, but unfortunately, approved treatments are only available for about 5% of those rare diseases. Researchers at the University of Pennsylvania and the University of Pennsylvania Orphan Disease Center have been making inroads for research and clinical aspects of rare diseases for more than 10 years. CDD is a genetic neurological disease. Well, more than 3,000 genetic conditions affect the neurological system, and the vast majority of them affect children. CDD has been one of the focus of Penn researchers together with the orphan disease centers for quite a while. My team is also researching other diseases, for example, Rett syndrome and Angelman syndrome currently, among many of the other primary neurological genetic diseases listed here. CDD is caused by a genetic defect. A large number of pathogenic mutations of the CDKL5 gene are known from affected patients. It's common for the vast majority of those mutations that no CDKL5 protein is detected in affected patients, or a highly truncated form of the CDKL5 protein, which is not functional anymore. So one of the approaches to overcome CDKL5 deficiency is restoring expression of the CDKL5 gene, so restoring normal levels of CDKL5 protein in the brain. And this is, in summary, what CDKL5 gene therapy is trying to accomplish. Before I describe to you our actual experiments and how CDKL5 gene therapy works, let's look at some of the molecular basis. The CDKL5 gene is normally encoded in DNA on the chromosomes in each cell. When the CDKL5 gene is expressed, it gives rise to an intermediate form called mRNA, which then gives rise to the fully functional folded protein, which is everywhere found in the cells. Now, if there's a CDKL5 mutation present on the DNA strands, RNA and ultimately protein cannot be produced. CDKL5 gene therapy now introduces a separate CDKL5 gene, also called the CDKL5 gene, into the cells. This CDKL5 gene coexists with the already present but mutated CDKL5 gene. So gene therapy is not changing the primary genetic code, it's not changing anything on the chromosomes, it simply puts there an additional gene to overcome the deficiency. This CDKL5 transgene now is expressed as mRNA and finally as protein, with the CDKL5 transgene protein being indistinguishable from the natural CDKL5 protein you would find in wild-type brains. Now, what is the process of gene therapy translational research? Well, in the beginning is the design process, where the treatment is generated and iterations optimized. followed by pharmacological studies, typically in mouse disease models, where one looks for disease correction and also to get already an early idea about potential transgene toxicity. Those studies are followed by toxicology studies, typically in primates, for example, monkeys. The main readout here is safety of the therapy, but those studies are also helpful for dose scaling and to optimize the route of administration. Ultimately, a clinical trial is conducted for first-in-human studies. Again, for phase 1 clinical trials, safety is the most important readout for the therapy, but also biomarkers are being looked at as an early indication of the potential therapeutic benefit. And those biomarkers can be either neurological tests performed in a hospital setting or by a neurologist, or rely on documentation by caregivers and families. The PEN-CD, the gene therapy program, is right now engaged in all the phases of the preclinical research, but not yet in the clinical phase. So let's start with the design and the early pharmacological studies in mouse models. We first went out and designed a DNA construct for CDKL5 expression. It is a rather complicated construct which encompasses the CDKL5 gene and regulatory elements. This is an iterative process optimized in cultured cells. Once this process is completed, the system needs to be somewhat modified to allow for delivery into animals. Gene therapy, in our case, relies on viral delivery to the animals or ultimately to the humans. We're using here an adeno-associated viral vector. This viral vector only contains the capsid, which is the outside structure of the adeno-associated virus, and is void of any of the other pathogenic genetic material. The CDKL5 gene now will be encapsulated in this AAV vector, and the AAV vector will be used to transfer the CDKL5 gene in the organism, in the cell, and ultimately in the nucleus to allow for the processes of CDKL5 protein transcription. It is important that those CDKL5 vectors are produced correctly and reliably using a bioreactor, and that they're highly purified, that they're suitable for animal use. For our animal experiments, we're using CDKL5 knockout mice, which express no CDKL5 protein whatsoever. The AAV CDKL5 vector was then injected directly into the CDKL5 knockout mouse brain. After 3 weeks, the mouse brain was harvested and processed for biochemical assays. One of the biochemical assays we're using was Western blotting for CDKL5 expression analysis. We found that CDKL5 was highly expressed in the CDKL5 knockout brain after gene therapy. As you see in blue as a comparison, black are the wild type levels you would find in those brains. We also used Western blotting to assess the activity of the CDKL5 protein. Again, we found significant activity of the CDKL5 protein after gene therapy. As you can see here in the blue bar, compared to the wild type levels, and then the somewhat not existing knockout levels. So that basically shows us that our CDKL5 vector restores CDKL5 protein expression and activity in the CDKL5 knockout mouse brain. Next, we slightly modified our assay to gain additional information about what happened in the mouse brain. Instead of processing the brain after collection for biochemical assays, it was fixed and then eventually cut into thin slices and mounted on microscope slides for microscopic observation. Before observing it with a microscope, the brain sections were exposed to fluorescence-labeled CDKL5 antibody, which selectively was detecting CDKL5 protein inside the cells. And as you can see here in the micrograph, the vast majority of the brain cells are expressing CDKL5 protein depicted in green. We quantified this in our mouse brains and found that at lower doses, we achieved about 20% of all the brain cells expressing CDKL5, whereas at a higher dose, it was an average of 60%. Next, we wondered what the effect of CDKL5 restoration in the knockout mouse brain would have. For this purpose, we performed the hind limb clasping test. It has been observed a long time ago that if you pick up wild-type mice on their tail base, the hind limb legs are splayed. Whereas in the CDKL5 knockout mice, they're crossed over and tucked under the belly. Now, when as before, we injected the CDKL5 knockout mice with the AAV CDKL5 construct in the brain, and in this case, waited for about 11 weeks before testing. As it has previously been found, there was virtually no clasping present in the wild-type mice as expected, and a significantly increased clasping index in the knockout mice. After treatment, we saw at low doses no significant improvements, shown in brown. At high dose CDKL5 gene therapy, we saw significant improving of the clasping phenotype. We also tested our CDKL5 gene therapy vector on wild-type brains. We did not see any effect after injecting wild-type brains with the CDKL5 gene expression vector, which shows that the treatment is safe. We conducted a variety of assays. Another assay among them is the open field activity test, where mice are placed into a plexiglass chamber. The plexiglass chamber is equipped with infrared crossbeams, and software is used to track the movement of the mouse within this array of infrared crossbeams over about half an hour. Again, we looked at wild-type mice, knockout mice, CDKL5 knockout mice, and CDKL5 knockout mice after gene therapy treatment. As it has been previously observed, wild-type mice have a continuous level of activity where they explore the open field chamber, whereas the knockout mice show a very pronounced hyperactivity over practically the entire 30 minutes observation time point. After gene therapy, we did not see significant reduction of this hyperactivity at the low dose. However, at the high dose, the hyperactivity was significantly reduced to almost wild-type levels. Again, when the gene therapy was injected into wild-type mice in light green and light blue, we did not see any change from wild-type behavior, again showing that the CDKL5 gene therapy is safe. In another line of experiments, we collaborated with Dr. Eric Marsh from the Children's Hospital of Philadelphia. Dr. Marsh is a neurologist who specializes in EEG measurements, and he found a way to record EEG from different parts of the mouse brain in a similar way like it would be done in a clinical setting. He had to overcome one obstacle. The currents, the electric brain currents in the mouse brain are so small that you cannot simply place a skull cap with electrodes on the mouse head like you would do in patients, but the electrodes had to be implanted in the brain structures to be examined. For the recording, then simply the electrodes were hooked up via a specialized connection with a recording equipment. Now, in preliminary studies, Dr. Marsh found that one aspect of the EEG spectrum, called the delta power, was significantly reduced in knockout mice, as you can see here in the red bar for knockout mice compared to the black bar for wild-type mice. After gene therapy treatment, shown here in dark blue, the delta power was restored to normal levels. Again, which is a further point showing that the treatment with CDKL5 gene therapy can restore normal brain function. Dr. Marsh is right now conducting follow-up studies where he also looks at the EEG measurements after gene therapy treatment when using visual and auditory stimuli, similar how it is done in the clinic. Okay, so I just described to you our in vitro studies and our early pharmacological studies with mouse models. Now I want to move on to talking about toxicology studies in monkeys and talking about aspects of safety, dose scaling, and optimizing the route of administration. When moving to primates, to monkeys, a number of considerations have to be made. For example, the mouse brain is fairly small, and so it's easily amenable for an injection to reach the entire brain with a gene therapy. The macaque brain is already multiple hundred times bigger than the mouse brain, and the human brain is even bigger than that. So now it turns out that a simple injection in the mouse brain is not sufficient to confirm gene therapy to the entire macaque brain. If one were to stick with the direct injections, multiple injections all over the brain would be needed, both in the macaque brain and in the human brain, to achieve a high level of transfer of gene therapy to the entire brain. We don't think that this is really a feasible method for administering gene therapy. Another method, which is straightforward because it's used in the clinical setting every day, is intravenous delivery. However, intravenous delivery of a gene therapy product, which is supposed to reach the brain, is highly inefficient, since the transfer of anything from the bloodstream into the brain tissue is inhibited by the blood-brain barrier, which means that most of the gene therapy you would inject IV never reaches the brain, but gets stuck in the blood capillaries. A way to directly reach the brain is injecting in the cerebrospinal fluid, or CSF. So the CSF is filling both the spinal column, and it is also filling several compartments inside the brain. It is outside. It is outside of the brain. The brain kind of swims in the CSF, and the CSF also flows along the major blood vessels. So with the CSF, you can get access to the brain. There's two ways to inject into the CSF. The one is via lumbar puncture, also called intrathecal administration, where a needle is directly inserted in the lumbar portion in the CSF, and that's clinical routine practice to administer a lot of drugs. Another way, which is maybe less common, but which is becoming more popular recently, is injection into the cisterna magna, or called ICM. The cisterna magna is a large cavity filled with CSF directly at the base of the skull, which is easily accessible from the outside by simply puncturing through the skin and the underlying membranes. Now, what are the initial targets for CNS gene therapy, which have already been tackled? Delivery to the spinal cord has been very successful for a variety of neuromuscular disorders, and is also highly efficient. That led, for example, to the development and the approval of Zolgensma, which is a AAV9 gene therapy for SMA, or spinal muscular atrophy. Direct AAV injections are also being developed in the clinic, however, more to treat specific cell populations, for example, for Parkinson's disease, where 1 or 2 injections could achieve the delivery of the AAV gene therapy vector to the cells of interest. Global brain delivery for primarily CNS diseases, unfortunately, is still somewhat inefficient. It is pursued in a number of clinical trials right now, primarily for lysosomal storage disorders. For the lysosomal storage disorders, the gene therapy relies on an effect called cross-correctional bystander effect. In this setting, the AAV vector is not only transducing the neurons as a primary event and expressing the gene therapy product here, but the gene therapy product, which in case of lysosomal storage diseases is an enzyme, is being secreted from those primary cells and now can affect the surrounding cells, thereby multiplying the effect of the primary transduction. Unfortunately, CDKL5 is not a naturally secreted protein, and so CDKL5 really has to be delivered to a quite large number of affected cells to restore the normal CDKL5 function in those neurons. A few words about IV delivery. So IV delivery theoretically provides broad access to the CNS to a dense network of capillaries. This has been shown in a mouse model. A few years ago, a group at Caltech developed a novel AAV capsid called PHBB, which allows very efficient delivery of a green fluorescence protein DNA to the mouse brain, which gives rise of green fluorescence protein expression all over the mouse brain, using very modest IV doses. However, when this was tried in non-human primates, unfortunately, this capsid is not able to perform the same way in non-human primates, and next to no AAV was crossing the blood-brain barrier using this capsid. Using the most popular CNS capsid called AAV9, that's the one which has been used successfully in Zolgensma. When that is administered IV, even at doses at high doses which are causing toxicity, it really only transduces a very modest number of neurons, and is mostly targeting actually non-neuronal cells. So AAV9 is really not very useful for IV injections to cure primarily brain disorders. However, the gene therapy program and many other groups are currently screening novel capsid for their capacity to cross the primate blood-brain barrier with high efficiency. So as it turned out, trying to do this line of research in mice was not very successful to translate it into primates. So right now everybody is focusing on primates and directly screening large libraries of capsid in primates for their characteristics to cross efficiently in the brain without causing any toxicity, and especially also using low doses, which would make such a therapy safe. So since this is all still in progress right now, CDKL5 gene therapy has been focusing on delivery through the CSF. To make advances for the CSF delivery, one of my colleagues recently conducted a study comparing the lumbar puncture administration with the cisterna magna administration. He was able to find that both administrations allowed a significant number of viral genomes to accumulate in the tissues. However, for brain, the ICM method was a lot more efficient than going through lumbar. For example, for spinal cord, the difference was not as big, but for brain, we believe it's a significant advantage to use the cisterna magna administration over lumbar puncture. That's why CDKL5 gene therapy at UPenn is focusing on developing cisterna magna administration. Now, in our studies with monkeys for CDKL5 gene therapy development at Penn, we've been focusing on macaques. Those are wild-type monkeys. They don't carry a CDKL5 mutation. Delivery was done through the cisterna magna, as I mentioned before, and the dose was scaled to brain weight from mouse to monkey. One group of the monkeys was monitored clinically for 2 months. We did not see any side effect or any adverse outcomes. A pathology review after autopsy was found that all tissues were completely normal. Our veterinary pathologist also focused on one particular peripheral tissue called the dorsal root ganglia and the white matter tracts along the spinal cord. In both of those tissues, toxicity for CNS gene therapies had been observed before for other indications than CDKL5. However, we were very happy to see that we saw absolutely no toxicity for CDKL5. So CDKL5 gene therapy is completely safe for the DRG and for the spinal cord, as reflected in very low severity scores, which are essentially within the normal spectrum you would find in a wild-type monkey. Thus, a main finding of our safety studies are the CDKL5 gene therapy is safe in non-human primates, and thus is also to be expected safe in a human clinical trial. So next, we looked at the distribution of the AAV vector throughout the monkey brain by counting the average number of the vector genomes in the particular tissues. As expected, the counts were quite high in both the DRG and the spinal cord. Those are the target tissues for therapies like Zolgensma, and we already know that the transduction here is very efficient. When looking at the brain, the transduction was somewhat lower, but we were very happy to see that both of our two vector construct we had tried out in the macaque monkeys were very efficient to transduce the cortex, in particular the very important motor cortex in the somatosensory cortex. Now, the next question is, now we have shown that there's presence of vector genomes, AAV genomes in the brain. So now, how does that relate to actually expressing of the CDKL5 transgene? So for expression of the CDKL5 transgene, we have to rely on looking at the RNA, because and you remember the RNA is the intermediate between the CDKL5 DNA and the CDKL5 protein, because the CDKL5 protein, which is expressed from the transgene, is indistinguishable from the CDKL5, which is already there in the normal monkey tissue. So the CDKL5 RNA assays RNA was quantified, and then we compared the amount of RNA we had in mouse brains, and those mice were injected with a higher dose, the therapeutic dose, compared to what we found in several structures of the monkey brain. RNA expression in the monkey brain was still somewhat lower than what we found in mouse, but we're very pleased to see that it had vastly improved compared to a previous iteration of our monkey delivery studies using a different AAV vector shown in green here. And so as you can see, especially the red AAV vector 2, what we call AAV vector 2, it is quite efficient in expressing RNA in brain, for example, in the motor cortex. Now, ongoing experiments address the key questions gating the transition into the clinical phase for CDKL5 gene therapy. The question is, what I had shown you, that is all overall measurements for taking a piece of tissue and then doing biochemical assays after mincing that tissue into small pieces and liquefying it. However, the question really is, how many brain cells do express CDKL5 in the end? And how many brain cells do we need to express CDKL5 transgene to expect the clinical benefit? We're pursuing two lines of experiments to address those questions. The one consists in careful dosing studies with CDKL5 knockout mice to relate the number of CDKL5 expressing brain cells after gene therapy with behavior and symptom improvement. And that will really tell us what is the threshold, what is the absolute minimum number of CDKL5 expressing cells a brain needs that it is functioning at least somewhat normal. So what's our lower level we need to achieve? And of course, we want to achieve a lot more than that, this lower level. And that comes to the second point, where we really need to do a more careful cell-based analysis in the monkey brain. So we are currently spending a lot of time doing molecular assays with the monkey brain and doing those on a single cell level, using advanced emerging sequencing, DNA sequencing and RNA sequencing technologies to really interrogate a single cell for which cell type, which of the many different neuronal cell types are expressing the CDKL5 transgene, and then at what levels is it expressed if you compare to the naturally occurring CDKL5. And we think really that answering those two questions are absolutely essential to convince ourselves that we are ready to apply for the permission to conduct a clinical trial for CDKL5 gene therapy. At the same time, preparations for clinical trial readiness are, of course, already underway. That is led independently by the Lulu Foundation. As you know, for example, natural history studies are conducted, and the Lulu Foundation has started to investigate clinical outcomes and clinical endpoints. And this work will be really key contribution for future clinical development of the CDKL5 gene therapy. Now, at the end of my presentation, I would like to summarize what we have achieved for developing CDKL5 gene therapy at UPenn so far. Well, we've developed an AAV vector for CDKL5 gene therapy. We've tested the CDKL5 gene therapy in a CDD mouse model for therapeutic efficacy. We found that CDKL5 protein expresses throughout the mouse brain and it is fully functional. We've conducted neurobehavioral studies with CDD mouse models, and we found that symptoms are significantly improved in the CDKL5 knockout mice after CDKL5 gene therapy. We then tested our CDKL5 gene therapy in macaque monkeys for safety. We optimized the brain transduction using ICM injection, and we did not observe any adverse events or any adverse pathology findings within 60 days in those monkeys. And we are currently optimizing CDKL5 gene therapy to express CDKL5 in the maximal possible number of brain cells to really maximize the probability for clinical benefit in a first-in-human trial. I would like to acknowledge the contribution of the many people who made this development of the CDKL5 gene therapy a success. First and foremost, of course, our director, Dr. Jim Wilson, for his leadership and his vision for this program. Then all the members in my translational research team, that's really the scientists who day in, day out carry out the experiments, take care of the animals. But all of those studies were also significantly supported by a number of core facilities within the gene therapy program. And last but not least, I would like to thank for financial support by the Lulu Foundation and Elage Bio, and all the good discussions both with members of the Lulu Foundation and the advisory board of Elage Bio. The gene therapy program is located at the edge of the idyllic University of Pennsylvania campus and close to downtown Philadelphia, which you can see in the background. Our labs are in a small 2-story building right at the foot of what we call the FMC Tower. So if you ever come to Philadelphia and the pandemic is, of course, is over, please do not hesitate to contact us and we will give you a tour of the building and tell you about some of the latest developments in the program. Thank you very much.